Imagine this. What would your reaction be if someone came to you and said, "Soon, computers will take over everything, from shopping to dating to stock market transactions," or even further, billions of people will be connected by some kind of network, and you will carry in your pocket a device far more powerful than the supercomputers of that time? You would probably have laughed out loud. But here it is, it's all come true. Science fiction became reality, and we've taken it for granted. Today we are at a similar point, but this time it's not about computers, it's about genetic engineering. Let's take a look at where this revolutionary story came from, what's being done now, and a groundbreaking development in 2020. Humans have been genetically engineering for thousands of years, but without knowing it. Through selective breeding, we have taken the traits in plants and animals that work for us and strengthened them. Bigger ears of wheat, stronger oxen, faster horses. But it took us a long time to understand how it worked, until we met DNA. DNA, deoxyribonucleic acid, is a complex molecule that governs all the processes of living things such as growth, development, and reproduction. Its structure contains the four basic letters A, T, G, and C. You can think of it as a biological alphabet. The order of these letters determines how a living being is shaped. Everything from your eye color to your hair type, and even your nose shape is hidden in these letters. Change the order of the letters and you have a different creature. Armed with this knowledge, scientists immediately started tinkering with DNA. In the 1960s, they used radiation to create random mutations in plants. The aim was to get a useful trait by chance. And yes, sometimes it worked. In 1970, they began to insert DNA fragments into bacteria, plants, and animals. During this period, the first genetically modified animal was born in the laboratory, a mouse. Even today, mice are one of the great protagonists of medical research. In 1980, genetic engineering became commercialized. From bacteria that clean up oil spills to the production of vital medicines, genetic technologies began to be used in many fields. For example, insulin is now produced in the laboratory, not from animals. In the 1990s, the first laboratory-produced food, the flavor-savored tomato, hit the shelves. This tomato had an extra gene added that delayed its rotting. What can we do today thanks to genetic engineering? Supermuscular pigs, fast-growing salmon, hairless chickens, glowing fish. Yes, you heard right. You can buy phosphorescent zebrafish for $10. We can do such impressive things. But there was a problem. Genetic editing was expensive, complex, and time-consuming until CRISPR came on the scene. CRISPR, opening a new era in genetics. CRISPR revolutionized the world of genetic engineering. It has reduced costs by 99% and made processes that used to take years can be completed in weeks. So how does this magical system work? CRISPR is actually inspired by a defense mechanism that bacteria have used for billions of years. Bacteria save the DNA of viruses, phages, that attack them in their archives. This archive is like a kind of memory card for the bacteria. When the virus attacks again, the bacteria use this memory to make RNA copies and weaponize a protein called Cas9. Cas9 cuts and neutralizes the invading DNA. Scientists have taken this system and adapted it to the lab. We now use this natural mechanism to make genetic edits. Thanks to CRISPR, we can make targeted gene edits, cut out harmful mutations, and insert desired genes. Is CRISPR a new hope for humanity? The impact of CRISPR is almost limitless. In medicine, we can treat genetic diseases like sickle cell anemia or cystic fibrosis. In agriculture, we can develop drought-tolerant or disease-resistant plants. In the environment, we can fight invasive species or bring back extinct species. But of course, with such great power comes great responsibility. What happens, for example, if genetic editing is used not only to cure diseases but also to create the ideal human being? Is it ethical to design children who change their eye color, run faster, or are smarter? These questions are likely to occupy humanity for years to come. The pandemic wasn't the only reason 2020 was memorable. This year marked a first in the world of genetic engineering. For the first time, the Nobel Prize in Chemistry was awarded to two women together, Emmanuelle Charpentier and Jennifer Doudna. What makes them special is that they developed the CRISPR-Cas9 technique. The two met at a conference in 2012 and quickly produced work that changed the rules of science. Usually, Nobel Prizes come after decades of research, and the team always includes several men, but this time things were different. By discovering CRISPR, Charpentier and Doudna gave the world of science a golden key. Today, CRISPR is used in every field from biology to medicine, agriculture to environmental sciences. CRISPR may sound like science fiction, but it is now a reality. Human beings have the ability to rewrite their genetic code. This is both exciting and frightening. In the right hands, CRISPR could make the world a better place. But if misused, it's hard to predict what will happen. So let's trust scientists, but let's also watch them. Because this new era will be the era of all of us, not just laboratories.